Hello everyone, welcome to Divya's BioWorld. In this channel, we will going to discuss about biological concepts which are useful for many competitive exams. So today we are going to discuss about phytochromes, one of the most important topic. Okay, phytochrome. It is a biological pigment. It is a biological pigment. So it is a blue light receptor. Blue light pigment. Okay, so light is not only a source of plant energy but also an environmental signal that instructs growth and behavior of plants. It is a blue protein pigment responsible for the perception of light into photophysiological processes. So it is a blue protein pigment, but it is a red light receptor. Okay, so it is a blue protein. It exists in blue color, but it receives red color. Okay, so it is a red light receptor. It is the only biological pigment present in reversible form. So it is only the biological pigment which exists in reversible form. That means it exists in two forms. One is PR form and another one is PFR form. So it exists in two forms. Okay, though, so these exist in reversible forms. So that process we call it as photoreversibility. Now we will discuss about that photoreversibility. Okay, so PR form. So when this PR form receives 630 nanometers of red light, it will convert into PFR form. Okay, when this PFR form receives 730 nanometers of light, that is far red light, it will convert into PR form. So mostly during the daytime we will be having the red light. So when PR form receives 630 nanometers of red light, that occurs during the daytime. So it mostly occurs during the daytime. Okay, so when PR form receives red light of 660 nanometers, it will convert into PR form, which mostly it occurs during the daytime. So when this PFR form receives far red light, that mostly occurs during the night times. So it mostly occurs during the night time. So when this PFR form receives far red light of 730 nanometers during the night time, it will convert into PR form. So during the night time, we know that most of the physiological processes are slowed down, right? So during the night time, why? Because all the physiological processes are slowed down why? Because due to this PR form. So we can call this as inactive form, and PFR form, which exists mostly during the daytime, which are responsible for many physiological processes, so we can call it as active form. Okay, so here we can say, so PR form, here it observes red light, so PR is red light observing form. And here PFR is a far red light observing light, right? So PFR is a far red light observing form. Okay, so PR when receives 630 nanometers of red light, it will convert into PFR form. And when PFR form when receives 730 nanometers of far red light, it will convert into PR form. Okay, so coming to the structure, phytochrome is a soluble chromoprotein with a molecular mass of 250 kilodaltons. Okay, so it is a soluble chromoprotein. It is a soluble chromoprotein and its molecular mass is 250 kilodaltons. It occurs as a homodimer made up of two subunits. It occurs in two subunits, each of 125 kilodaltons. Okay, it exists in two. So it is a homodimer. Homodimer means two monomers combine to form one combine to form one homodimer. Okay, so each monomer weight is 120, so both combined it is 250 kilodaltons. Okay, next, Each subunit consists of two components. Each of so Each subunit consists of two components: light absorbing pigment molecule, that is chromophore, and a polypeptide chain, that is apoprotein, which initiates signal transduction pathway. So here we can see this is the structure. So it is a two monomers. So if we take one monomer consists of one chromophore. It is a light absorbing pigment, that we call this form as chromophore, and this is apoprotein. Okay, this is apoprotein. Chromophore and apoprotein, two subunits. One monomer consists of two subunits. One is chromophore and another one is apoprotein. Okay, so chromophore is a light absorbing pigment, and apoprotein, both combine to form both combine to absorb the light. Okay, chromophore is a linear tetrapyrrole ring. Linear tetrapyrrole rings. It consists of linear tetrapyrrole. Tetra means four rings. So four pyrrole rings. Combine rings attached to the protein to thioether linkage. Thioether linkage means both combined with the help of sulfur. Okay, thioether linkage to a cysteine residue. So chromophore and apoprotein combine with thioether linkage to form to a cysteine residue. Okay, and so we have said that it exists in PR form and PFR form, right? So the main difference in the structure is, in PR form it exists in cis form, and in PFR form it exists in trans form. Okay, this is PFR form and this is PR form. So here it exists in cis form, right? And when they receive red light of 630 nanometers, it will convert into transform. This is transform. This is transform and this is cis form. So when red light of 630 nanometers, if it observes, this cis form will isomerize to transform. So we can say cis form is activated form, and transform is activated form, and cis form is inactivated form. The principal difference between the PR chromophore and PFR chromophore appears to be a cis trans isomerization of the methane bridge between rings C and D. So here these are C and 2 rings, right? In the methane bridge, this is a methane bridge. So only the cis and trans conformations is the mainly difference between PR and PFR form. Okay. Next, coming to the functions. So it helps in photomorphogenesis, the process by which plant development is controlled by the light. So based on the light, the plant growth and development will be depends. Okay, and germination. PR form helps in germination. So we say PFR form is the active form. So PFR form helps in germination. De-etiolation. So de-etiolation means the seedlings which are grown in dark conditions, so it helps in de-etiolation, opposite reaction. Okay, and inhibition of stem and petiole elongation. So next, chloroplast development and chlorophyll biosynthesis, and photoperiodism. So they play major role in photoperiodism. So photoperiodism is the biological phenomenon in which a plant responds to relative lengths of day and night to regulate developmental process, most notably flowering. Okay, so photoperiodism is the plant response to creative day length of light, especially for flowering process. Okay, so we know, right, long day plants means they require more daylight, and short day plants means they require very short length of light. Okay, So long day plants when receive red light, it will convert into PFR form and helps in flowering. So here long day plants need more day length, right? Compared to the critical day length, they need more daylight. So when they receive long day plants, when they receive red light, it will convert into PFR form and it will starts flowering. But in short day plants, when they receive far red light, it will convert into PR form and helps in flowering. So if you observe long day plants, when they receive red light, induces flowering. Short day plants, when they receive far red light, it will induce flowering. But if the short day plants receive red light, they will inhibit flowering. Okay, it is a major difference between them. Okay. Next, coming to the discovery, Bothwick and Hendricks in 1932 showed that red light of 613 to 680 nanometers elicits germination in lettuce seeds, whereas far red light inhibits the process. So they observed that in lettuce seeds, when they receive red light of 630 to 680 nanometers, it promotes germination, but when they receive far red light, that is 730 to 740 nanometers, it inhibits the process. So they later Butler in 1959, firstly they have isolated it from etiolated oat coleoptile and used the term as phytochrome. Okay. And localization, if we observe, if we like we need to know, right, where the phytochrome will exist. So distributed in plant kingdom, it is distributed almost in plant kingdom, detected in monocots, dicots, gymnosperms, ferns, mosses and algae. Detected in different parts of the plant. It exists in nucleus, cytosol, and highest levels are found in etiolated seedlings, especially in the meristematic regions. So in etiolated seedlings, mostly PR form will they. Okay. And so mostly this phytochrome biosynthesis will start in nucleus, and proper development will takes place in cytosol. Okay. Thanks for watching. Hope you understand. If you have any doubts, you can comment below. Thank you.